Hello everyone, hello ma'am. My name is K.S.G. Hinobatin and this is our report and distinguish the major features of glycolysis, Krebs cycles, electron transport chain, and osmosis. Glycolysis is the first step of cellular respiration, which starts with a single glucose of molecules. Glycolysis takes place in the cytoplasms of the cell, where ATP donates two phosphates to glucose in a process called phosphorylation. Phosphorylation is when phosphates is added to two molecules. Glucose is broken into two 3-carbon molecules containing phosphate groups that donated by ATP molecules. Next, the phosphate groups removes 3-carbon molecules in a chemical reaction chain that produces 2 ATP from each molecule. But during the process of removing the phosphate group from these carbon molecules, an electron is stored in a molecule called NADH. When NAD plus takes an electron, it becomes NADH. In the same way that you can put money in the bank so that you can use it later. Glycolysis stores energy in a form electrons inside in NADH. The two 3-carbon molecules in the end of the glycolysis are known as pyruvate. Glycolysis, which is the first step of cellular respirations, doesn't require any oxygen, which means it's anaerobic, meaning without oxygen. Glycolysis produces not increase 2 ATP, stores 2 electrons and 2 NADH molecules. To have a further learning of glycolysis, we have to watch this video.
Hey, it's Professor Dave. Let's talk about glycolysis.
He knows a lot about the science stuff. Professor Dave explains.
We need energy to do literally anything, from running a race to simply breathing. And every cell in your body is furiously producing all this energy all the time. Where does this energy come from? Well, technically it arrives from the sun. The sun is releasing enormous amounts of energy as a byproduct of nuclear fusion reactions, and that energy makes its way to Earth where it can be absorbed by plants. Through photosynthesis, which we will discuss later, plants use sunlight as well as carbon dioxide and water to make glucose. And it is all of this glucose, among other biomolecules, that becomes the starting material for metabolic processes in our bodies. This degradation of biomolecules to generate energy that cells can use is called cellular respiration, or sometimes more specifically, aerobic respiration. Let's see how this works, looking at glucose as the substrate. Aerobic respiration requires oxygen, so any organism that breathes in oxygen from the atmosphere is doing so in order to facilitate aerobic respiration. Glucose, which we can either consume as starch or break off from glycogen stored in the cell, can be converted through metabolic pathways in the presence of oxygen into carbon dioxide, which we breathe out, water, which is most of what we are, and energy, the energy we need to think and move. This is not unlike the combustion reactions that happen in an engine, so it's a reasonable analogy to view biological organisms as machines. The electron exchanges that occur throughout these metabolic pathways utilize the electron carrier NAD+ and its other form, NADH. This is a dinucleotide with an interesting base, nicotinamide, that can exist either as NAD+ with a positively charged nitrogen atom, or, if reduced, it can become NADH. This transfer, facilitated by an enzyme called dehydrogenase, helps catalyze the breakdown of glucose. Cellular respiration happens over 3 major pathways. There's glycolysis, the citric acid cycle, and oxidative phosphorylation. Let's focus on these one at a time. Glycolysis comes first, and it happens in the cytoplasm of the cell. This is the process by which glucose molecules are split into 2 pieces called pyruvate. This first pathway is actually anaerobic, meaning it does not require oxygen, so it is the most evolutionarily ancient metabolic pathway, occurring in even the simplest cells. In this pathway, one glucose molecule can yield a net of 2 ATPs. It requires 10 enzymes to happen, which catalyze each of the 10 steps, as well as an investment of 2 ATP molecules in the preparatory phase to get 4 ATPs back over several steps in the payoff phase. The names and details of each individual reaction may not be of interest to every viewer, but in case they do interest you, here is a brief overview of each step. First, the hexokinase reaction. The enzyme hexokinase phosphorylates the oxygen on carbon 6 to make glucose 6-phosphate. The polar phosphate group traps the molecule inside the cell and also reduces the concentration of regular glucose inside the cell, which encourages more glucose to enter by diffusion. This step costs 1 ATP, which provides the necessary phosphate group for the reaction. Next, glucose 6-phosphate isomerizes to become fructose 6-phosphate, a process which is catalyzed by phosphoglucose isomerase. After that is another phosphorylation, this time on the carbon 1 hydroxyl, which gives us fructose 1,6-bisphosphate. This step is catalyzed by phosphofructokinase 1, and it will cost another ATP. Now, this molecule is ready to be cleaved into 2 smaller ones. Fructose bisphosphate aldolase is a lyase enzyme that will split fructose 1,6-bisphosphate into a molecule of glyceraldehyde 3-phosphate, or GADP, and a molecule of dihydroxyacetone phosphate, or DHAP. The DHAP will be converted into another molecule of GADP by the enzyme triose phosphate isomerase, which leaves us with 2 molecules of GADP. That's the end of the 5-step preparatory phase, with 2 ATPs spent to achieve the 2 phosphorylations. Now it's time for the payoff phase. Let's just look at one of our 2 GADP molecules from the preparatory phase, and we see that the first thing that will happen is an oxidation to become 1,3-bisphosphoglycerate. This requires NAD+ and a free phosphate, or inorganic phosphate, to occur, and the enzyme involved is called glyceraldehyde phosphate dehydrogenase. Next, a phosphoglycerate kinase will catalyze transfer of a phosphate group to ADP to become 3-phosphoglycerate, producing 1 ATP in the process. Since each of the 2 GADP molecules will make 1 ATP, that's a total of 2 ATPs for half the total payoff of glycolysis. Then, phosphoglycerate mutase transfers the remaining phosphate from this hydroxyl to the next one over to make 2-phosphoglycerate. Then, enolase catalyzes a dehydration, resulting in the loss of this hydroxyl group, which will produce phosphoenol pyruvate. And lastly, the remaining phosphate group is transferred to an ADP by pyruvate kinase, generating another ATP and the pyruvate we discussed before. So altogether, it's a 10-step process. The first 5 steps comprise the preparatory phase, which take 1 molecule of glucose and produce 2 molecules of GADP. This will cost 2 ATP. Then, the other 5 steps make up the payoff phase, in which each molecule of GADP will be converted into pyruvate, producing 2 ATP each in the process, for a total of 4, meaning the net energy production from 1 molecule of glucose is 2 ATP. If you are required to memorize basic facts about glycolysis, this should probably suffice. But if you wish to memorize this process in more detail, here is a table that lists the necessary enzymes for each step, as well as any relevant input or output besides the actual substrate. Certainly, the main thing to remember is that in glycolysis, glucose in the cytoplasm of the cell is converted into pyruvate, which will then move on to the next stage of cellular respiration.